Up now with our PowerPoint on scientific method. We've already covered the first three steps of the scientific method. We've talked about number 1, ask a question, number 2, gather information, and number 3, which is to form a hypothesis. So step 4 now is to do an experiment. Now, when you're doing the experiment, you have to ask yourself, how can we find the answer? How can we know whether or not the answer to our hypothesis is correct? So what specific steps are we going to follow in order to draw a definitive conclusion? Whatever those steps are, we need to make sure that all experiments meet 4 criteria. Number 1, does it answer the question? Number 2, is it repeatable? Number 3, does it have a control group and a variable group? And number 4, does it reduce or eliminate error? Now, let's look at each one of those 4 criteria a little bit closer. So first of all, does it answer the question? Your data needs to support the conclusion. Whatever that conclusion is, you have to have data to back it up. Now, there's 2 types of data. There's qualitative data, which are those things that are based on characteristics, qualities of an object, like tall, brown, healthy, etc. And then there's quantitative data, which is, that's what we typically think of as data, the hard facts, the statistics. Those are numbers. So 15 centimeters, or 1 milliliter, or 1.234 grams per milliliter, etc. But in order to be able to answer the question, you need to have data to support the conclusion. Is your experiment repeatable? In order for your experiment to be repeated by others in the exact same way as you have performed it, you need to be specific about everything. This is the one thing that all scientists need to improve on. You need to write down every single little mundane detail. You have to dot your I's and cross your T's. There should be no ambiguity in your directions, which means I should not be able to interpret your directions in any way other than the exact thing that you meant. So 2 people from different parts of the world should be able to read the directions and do the exact same thing as you have instructed them to. Make sure you're specific. Now, the 3rd criteria was you have to have a control group and a variable group. The control group is the group that is the basis for comparison. So your results from the variable group are compared to the results of the control group to determine if the variable is responsible for any change. For example, plants are given the exact same treatment, which means you have plants that get fertilizer and you have plants that don't. The plants that don't get fertilizer are the control group. They get everything else that the plants with fertilizer get, except they don't get the fertilizer. So the independent variable is the one thing that you change relative to the control group, and you can only have one independent variable. For example, that's the plants with fertilizer. Now, the dependent variable that's the stuff that changes as a result of the independent variable. That's the thing that you measure. So, for example, we're going to measure plant height. Okay. So if we just take a look at this slide one more time, we have the control group, which is the group of plants that have water, sunlight, dirt, everything is the same, as is the group with fertilizer. The independent variable group has fertilizer, the control group does not have fertilizer. Otherwise, they are the exact same. The dependent variable is the thing that we are going to measure on both the control group and the independent variable group. So we're going to measure plant height from the control group to see if that's different than the plant height with the group with fertilizer. And the last slide here on criteria is the type of error. There are 2 types of error. There's systematic error, which means something is wrong with your system. You can eliminate this type of error, and it stems from incomplete instruction. So if you do things incorrectly, in the wrong order, whatever, that's systematic error. You can fix systematic error, and you should be able to diagnose systematic errors. Those things should be repaired in the lab and should be eliminated from all of your experiments. And the last thing, of course, then is random error. Random error happens because we are human. Humans make mistakes. You cannot eliminate random errors, but you can reduce them significantly by practicing good lab technique and double-checking your work. Now we'll just kind of run through a couple systematic versus random errors that you can try on your own. We measured wrong. That is a systematic error. If you measure wrong, you should go back and fix it. We might not have seen exactly where the marshmallow landed. That would be a random error. because you probably will not be able to see exactly where a marshmallow landed. We calculated wrong. Again, that's systematic. The beaker cracked. That's systematic. The thermometer was only marked in 1 degree increments, so we had to estimate the temperature if the line was between the marks. That would be random. Okay, and Mr. Boyd didn't time us correctly. That, of course, would be systematic. And then you can read the rest and determine whether or not they are systematic or random errors. Remember, systematic errors should not exist in your lab, and if they do, then you did the lab incorrectly. Random errors should be discussed in the conclusion, but will probably not have a significant impact on the overall results. The next step of your experiment is to analyze the results. We can organize our data so it informs us about our hypotheses, and we can do so using graphs, charts, data tables, etc. Now, when we talk about graphing, we have different types. We have a bar graph, which is used to show independent data. We have line graphs, which is used to show trends over time. And then we have pie graphs, which are used to show percentages of a whole. Generally speaking, in science class, you are going to use a line graph. Most of the time, probably 90% of the time, you are going to use a line graph to show your trends over time. It's important, though, when you're doing a graph that you do so correctly. On the left here, you see all the information that needs to be included on the graph. First of all, the title. It needs to have a title that is indicative of the information, that it's informing the audience about what information is contained in the graph. You need to have a key if necessary. You know, if you have more than one line, you need to have a key. There only happens to be one line here. The axes need to be labeled. So you can see here that the y-axis is labeled temperature, and then in parentheses is the unit, so degrees Celsius. The x-axis is labeled time, and in parentheses then we have the unit minutes. Scale. Scale just refers to having a breakup of the numbers that is consistent, one that is aesthetically pleasing, one that works within the graph. Of course, you need to have your data. You need to actually have a line on your graph, or bars on your bar graph, or definitions in your pie graph, whatever the case is. And then lastly, it's probably the most important, but your independent variable goes on the x-axis. So this is the thing on the x-axis that you have control over. You have control in this example of time. You have control over how long this substance stays in the heat or whatever. The thing that you cannot control is the dependent variable, which is the temperature of the substance. So you can control how long it stays in there, but you cannot control the temperature of that substance. So this is the independent variable. This is the dependent variable. You must remember that the independent variable goes on the x-axis and the dependent variable goes on the y-axis. Lastly, draw a conclusion. So, did it work? And was your hypothesis correct? Why or why not? And then the seventh step is to communicate the results, and that is because science is a communal process, which means we benefit from the work of other people. We don't just kind of maintain our own little solitary existence within a lab and just hope that people learn about us from reading or whatever. We like to go out and talk to people and report our results to other scientists. And there's a process called peer review. So other scientists need to basically sign off on the work that I've done or the work that others do and say, yes, it is valid, it is good science. And scientists communicate using professional journals. We, however, are going to communicate using lab reports. And a simplified lab report is called a rerun. Rerun is an acronym that stands for restate what you did, explain the purpose of the experiment, state your results, tell me about your uncertainties, so why might you doubt your results, and then tell me new things that you've learned. What did you learn as a result of this activity? Now, let's take you through the 7 steps of the scientific method and with our example of how does fertilizer affect plant growth. So we're going to ask a question, how does fertilizer affect plant growth? The information that we're going to gather is the effect of water, fertilizer, sunlight, etc. on plant growth. Our hypothesis says that adding fertilizer to a potted plant will increase the height of the plant because fertilizer provides the nutrients a plant needs to grow successfully. Experiment is how specifically are you going to test this hypothesis in a way such that the fertilizer can be the only reason for the difference in plant height. and we're going to analyze the results, make a graph of our data. In this case, I would choose a line graph, one that showed the amount of fertilizer on the x-axis and then the height of the plants on the y-axis. I'm going to draw conclusions, and then I would communicate my results with a rerun.